certainly can raise your hand or gently interrupt, and Adam can answer your questions during the talk. But if it can wait, we'll certainly have a question answer session at the end. And I believe the committee will just take a few minutes on our own to discuss, and then we'll be done. Okay. So here's what we'll do. It's 11:06. Any news on Slack or email? I haven't seen anything on text yet.
No.
I'm just going to tell him to join when possible. I'll tell him we'll get started.
Yeah, maybe something came up, I think.
No problem. Yeah. Okay, so Adam, I'm going to do an introduction. You can let people in if you see anybody as you introduce. So thank you all of you for coming. Welcome to friends and family and the parents of Adam. I had to think about what I'm going to say for this introduction. So I was like looking back at all the different things that Adam has done since he joined. So there's a lot to say, but I'll try to make it sort of comprehensive yet brief. So Adam came to my office, I would say about 6 years ago, interested in rotation. He came from UCSD, one of my own colleagues when I was a PhD student, Dr. Todd Coleman, who's a professor now at Stanford, had worked with Adams and wrote a letter and called me and told me all about Adam. So I was very excited to have him rotate in my lab. He also did a rotation in Dr. Vogelstein's lab. So I was a little worried because Adam's on the very high-tech side and I'm very low-tech. And so I thought, okay, he's going to go with Josh because Josh is everything, high-tech, low-tech, everything. But he didn't. And I'm so happy he didn't. And Josh is so annoyed and sad that he didn't because Josh then served on his committee and has seen all the things that Adam has done. So let me just tell you a little bit about Adam and his journey here. So, he's one of the few students that has multiple fellowships under his belt. So he's a National Science Foundation Graduate Research Fellow. He won that award. He's a Whitaker Fellow. He's a Chateaubriand Fellow. The Whitaker allowed him to actually travel to Marseille over a period of time where he worked with Dr. Victor Gers on the Virtual Brain Project. He's also an ARCS chapter fellow. So he's been well supportive all on his own through all these various fellowships, which are incredibly difficult to get, just one of them, but he's gotten multiple, and including a fellowship for his postdoctoral. So now he has received 2 years more of funding all on his own. So Adam has, I would say, strengths in multiple dimensions way beyond his years. You'll see through his research, he's a brilliant and bold researcher. I think the problem he tackled is a difficult problem. It's a clinical problem, and you'll see some of the clinicians on the call today, and Dr. Crone, epidemiologist, neurologist here, research leader in his field at Johns Hopkins, has served as his on his thesis committee, and very much as someone who has driven, the clinical need and really carefully looked at the research that Adam has been doing. He's a determined translator. So the work that you're going to see today, we co-founded a startup company together called Neurologic Solutions. And his solution that he's going to talk about today, his algorithm, has been FDA cleared. So, and Adam led that whole effort through the whole FDA submission process. This is just really, really rare for a PhD student to get his or her research FDA cleared before defense. So congratulations on that. He's also done a lot of things outside of the classroom, more outreach. So I know that he founded an organization, I believe at UCSD, called the Engineering World Health, EWH. And he received funding from the Clinton Foundation. And EWH's mission is to develop cost-efficient solutions that bring modern medical technologies to rural hospitals in underdeveloped countries. So this is just to have you understand that everything he's done, and you're going to see the research, the translation, is not just what defines Adam. He really goes out there, in terms of doing more outside. And that's just some. I know, Adam, you've done things that I don't even know about, and I hear about it later, and I'm like, oh my God, that too. So very much a big multitasker. So super excited to introduce you today. It's been an honor being your PhD mentor. I'm so excited that you're still going to be an entrepreneur and work with me on this technology, your work, as well as others. Hopefully we'll build a big company, and hopefully you'll still do big things as a professor, bringing more research and translating that work to really, really improve clinical treatment, diagnostics, and outcomes. So with that, take it away, Adam.
Hey, yeah, thank you so much for those kind words. And yeah, good morning to everyone, friends and family, collaborators and mentors that are able to join me today in this talk and celebration. Yeah, thank you all. I know it's a little bit early for those on the West Coast, but yeah, we'll try to make things succinct and on time. So today I'm going to be talking about localization of the epileptogenic zone in drug-resistant epilepsy, providing a dynamical systems perspective. And I'm a biomedical engineering PhD candidate advised by Dr. Sri Sarma, Dr. Nathan Krohn, and Dr. Joshua Vogelstein. And so my research addresses a clinical problem in epilepsy. Currently, over 65 million people suffer from this chronic condition. And actually, a life-changing surgical treatment is available for clinicians where if they can remove this region called the epileptogenic zone, or EZ, in the patient's brain, then the patient will be seizure-free. And so they can do this process. However, for drug-resistant epilepsy patients, they actually have pretty poor surgical outcomes, averaging around 50% success. And as a result, they lose money on each failure, and only 18% approximately even choose to offer treatment based on this poor outlook. The EZ is not easy to localize because it's defined retrospectively. And it's defined as, quote, the brain tissue needed to be removed in order for the patient to be seizure-free. And this sort of definition presents some challenges in treatment because it doesn't tell you what to look for prospectively. Modern theories believe the EZ to be an area of cortex responsible for the generation of clinical seizures. And in this sense, it can be embedded within a brain network that involves normal healthy brain regions and even propagation regions. Typically, clinicians implant electrodes in an attempt to locate this region. And if their hypothesis overlaps with the true onset zone, then the patient will be seizure-free. However, because the EZ is not well-defined, as I said, then this localization might be inadequate, and then subsequent surgical excision will not render the patient seizure-free, even with some brain tissue being removed permanently. Localizing the EZ clinically generally involves invasive brain surgery, lengthy electrode activity monitoring, and a large clinical team to inspect various EEG signatures or various EEG recordings for various EEG signatures. And then this motivates the need to identify the EZ from EEG recordings reliably that can then in turn define the EZ prospectively. However, because no biomarker is available, people have tried various approaches to search for EEG markers and to define the EZ prospectively. For example, one prior approach for an EEG marker is the power within a frequency band. So the power in a certain frequency range might be used to quantify the likelihood that a specific brain region is part of this epileptic network. For example, one might look at the gamma frequency band, which is defined between around 30 to 90 hertz. And they might look at this for various EEG channels. However, even though it describes certain epilepsy phenomena that is apparent, it doesn't actually capture the network nature of epilepsy and isn't sufficient to separate EZ from non-epileptic regions. And as such, no one clinically relies on the spectral features alone to prospectively define the EZ. Another prior approach is known as high frequency oscillations, or HFOs. And this was and is an exciting area of research currently because they can be defined interictally. That is, they occur between seizure events and don't require you to observe, an actual clinical seizure occurring. Although there's no unified definition, they're basically short oscillations that occur at very high frequencies and are isolated to, a subset of different channels. However, even physiological HFOs exist, where there were different studies that showed this. And so this calls into question, what is physiological versus a pathological HFO? And this question remains to be answered. And a Google Scholar search will show an explosion of research in this research area of HFOs applied to epilepsy. And many researchers have tried to correlate HFOs to the seizure regions. And there's even prospective clinical trials, but so far, most of them are inconclusive to this date. Another common approach is to interpret the functional epileptic network as a graph. So one can take EEG data, compute pairwise correlations between all pairs of electrodes, and interpret that as a functional connectivity of the brain. And then one might be interested in computing various graph metrics, such as the average shortest path between two different nodes or two different electrode EEG electrodes. When analyzing a graph, one might consider this graph metric and apply it to localize the EZ. Now, when we consider graphs with 10 nodes and 20 edges, one can have a variety of different values of this metric, as shown here. However, by just varying 2 to 3 edges on this graph, one can vary the graph metric substantially within this subset of ranges. And as a result, one can apply this, and Chun et al. Actually showed this for a variety of different graph metrics. And so what does this mean? It means that when we don't actually have observations of the underlying graph structure and try to estimate this from data, incorrect estimation can lead to misleading downstream graph metrics. And when applied to easy localization, can be misleading. And so moving forward, how can we actually improve the state of localization in these patients? Well, we talked about 3 different bucketed prior approaches, and they each have some pros and some cons, of course. So moving forward, we want to really leverage all these into and also incorporate our biological understanding of the epileptogenic zone and create a dynamical network-based representation. And so how will we do this? So first off, just rather than looking at the problem from a static graph approach, as was previously mentioned, we actually approach it from the lens of a dynamical system. So that means incorporating our understanding that, the brain is a dynamical, organism, essentially. So our first observation is that the EZ is fundamentally unstable. That is, small perturbations to the brain lead to actual seizures occurring in the patient. For example, in a healthy brain, the brain is not seizing and is a stable and balanced system. In a seizing brain, the brain is an unstable and unbalanced system. Our second observation is that these imbalances can actually occur due to changes in neural connectivity between different populations of neurons. For example, De Felipe et al. Shows that the loss of chandelier cells decreased damping, which caused the network to become epileptic. Jin et al. Showed that the abnormal axonal sprouting increases excitation, which also leads to the network becoming epileptic. Taken together, these result in an unbalanced brain network that is sensitive to perturbations that can then in turn generate clinical seizures. So taking these 2 observations and our basic understanding of the epileptogenic zone from biology, we coin a term, neurofragility, which looks at the smallest amount of connectivity change that would actually result in a network imbalance. And so our goal is to compute this exactly. And so, for example, if we have a set of EEG implantations, then the neural fragility of a specific node or specific electrode is equal to the smallest amount of change in its connections to its neighbors that will cause the overall network to become imbalanced and capable of presenting with seizures. So moving forward, we're going to demonstrate how to compute this actually. So our hypothesis is that we believe that EZ leads to this unbalanced EEG network that's an implicit imbalance between excitation and inhibition. And so we hypothesized that the EZ corresponds then to the most fragile regions of the network, where fragility will be computed in the following sense using an algorithm. So in the rest of the talk, I'm going to first develop an algorithm that computes neurofragility from data. And then subsequently, we're going to perform a retrospective study and show you the results. And then finally, we're going to apply this data on some rare interoperative recordings that will further validate neurofragility as a marker for the EZ. So to start off, we want to develop our algorithm. We're going to model the EEG network as a linear time varying system. There's a lot of math involved, but our intent is essentially to formulate the problem to arrive at a least squares problem that can then robustly estimate our linear system and efficiently. We then demonstrate that this model captures dynamics across the entire network within small windows of time. So for example, here we have an actual EEG clip that's from a real recording. And then here's a simulated EEG clip of the same window using our estimated system. Now, when we look at the real and simulated EEG overlaid during interictal, preictal, and seizure periods, that is different periods that the brain is either seizing or not seizing, we see that the model is still a relatively good fit across a variety of different electrodes. And so this is good to see, because now we can estimate our dynamical system. We can next ask the question, how sensitive are various EEG nodes to perturbations? That is, how can we quantify neurofragility? So we can actually compute this, and we can actually quantify this explicitly all from data. And so then fragile nodes in this network have the smallest delta term. So that means they're the most sensitive to perturbations in this sense, and we can compute this exactly from data. Now, hopefully I didn't lose you there. So here I'm going to walk through a very simple two-node example. So starting from the left to the right, we have an EEG network model. So there's some math involved, but maybe you're more comfortable with the second column where we have a graphical depiction of just excitatory and inhibitory node. And then the third column shows our perturbation. And then the fourth column is where we apply an impulse response to node I. We can observe what happens in response. So here we see some transient-like activity, but both nodes' activity returns to a baseline. So that is, this is still a stable system. Now we can imagine perturbing the node I's connectivity to its neighbors in such a way with a magnitude of square root of 5. And then we can apply this impulse response again. And then we can observe, similarly to the above, transient-like activity, although a little bit different, but it still is a stable system. Finally, if we perturb nodes I's connectivity in just the right amount, that is in this case, square root of 8 magnitude, and we apply this impulse response again, we can observe a bifurcation or a change in the system's behavior. And that is, the system is actually now unbalanced. So in order to compute a neurofragility heat map from actual EEG implantation, we'll take a small sliding window of data and estimate the system as described earlier. We're going to then compute fragility for every single EEG channel in that window and normalize such that high values indicate fragile regions and low values indicate non-fragile regions, where red is high, blue is low. We can then repeat this process in the next time window and compute the next window for neurofragility for every single EEG channel. We can then repeat this process again. And then to summarize, we can go from an EEG snapshot and a sliding window of time to a spatial temporal neurofragility heat map that shows electrodes on the y-axis over time on the x-axis, and the color represents the evolution of this neurofragility values over time, normalized across all electrodes, where red indicates, again, more fragile and blue indicates less fragile. So next we sought to collect some data. So some EEG data that's common for invasive surgery, known as ECOG and SEG, where they're either surface on the brain or depth electrodes. And so we collected, 1 to 3 snapshots for seizures, different outcome data, clinical etiology, the clinical hypothesized EZ, or otherwise known as the seizure onset zone, different demographic data, and other patient metadata. This results in 91 subjects across 5 different clinical centers, Cleveland Clinic, NIH, Hopkins, University of Maryland, and University of Miami. We also converted the data to community supported format that facilitates trivial data sharing and analysis. To accomplish this, I became a developer in these 3 organizations that are dedicated to open science. And then we made this actually data set as open access as possible, available on an NIH supported platform called Open Neuro. So now that we have our data set and algorithm, I'm going to present to you our retrospective study that actually validates this neurofragility as a biomarker for the EZ. So first off, we're going to look at a representative example from a patient at the NIH, or National Institute of Health, with a successful surgical outcome, meaning that the clinicians hypothesized epileptic region was correct in that the patient became seizure-free. And so here on the left-hand side, we have a schematic of their implantation, and the red highlighted electrodes indicate what the clinicians hypothesized. The orange overlap indicates where they resected. If we look at a raw EG map with the electrodes highlighted in red on the y-axis, indicating, again, the clinician's hypothesis, time lock to seizure onset marked by the black dashed line. You know, even for me, someone that's not visually or not trained as an epileptologist, I can definitely see some activity indicating, like, that makes me aware that, okay, this is where the clinician's hypothesis was epileptic. If we look at an exact match, same window of time for neurofragility heat map, with, again, the electrodes marked in red on the y-axis indicating the clinical hypothesis, we see the heat map evolution after the black dash line. We see, various fragile regions and fragile activity. Interestingly enough, we actually also see that ATT1, which was a highly involved or highly discussed clinical region, was also highly fragile before seizure onset, indicating maybe neurofragility can be used intricately, that is, between seizures, to locate the EZ. And so this is a good case to see because this patient was seizure-free, and so we overlapped with the clinical hypothesis relatively well. Next, if we look at a patient with a failed surgical outcome, again, the red electrodes indicate the clinical hypothesis, the orange indicates the estimated surgical resection. If we look again at these two, a raw EG map, as well as a neurofragility heat map, this time, the clinical hypothesis, looking at the raw EG, it's a little bit harder to distinguish what is epileptic versus not. But that's only for me because most of us are probably not trained as EG readers. Now, if we look at the fragility heat map, we see a few things. One is that there's not as much fragile activity in the red, in the clinical hypothesized regions, but there's also highly fragile regions outside of the clinical hypothesis, indicating that potentially clinicians didn't resect fully the epileptogenic region, and that's maybe why the patient was not seizure-free afterwards. So in our retrospective study, we sought to quantify how neurofragility can be used as a biomarker, and we want to quantify and validate how effective this is. And so first off, we take a look at how clinicians localize again. So they take several snapshots of EEG from seizure events. They combine this in such a way using their own knowledge based on neuroimaging and different patient evaluations. And they combine this with a team to form a consensus in their confidence in localizing what's the seizure onset zone. And then they use this clinical hypothesis to then predict, okay, where is the EZ? And then we can perform subsequent surgery and measure the outcome. In a similar sense, we want to mirror this approach in our experimental design, computational experimental design. So we take those EEG snapshots, we compute neurofragility heat maps, And we're going to split up the data into one, the clinical hypothesis, and two, the SOZ complement or SOZ-C, which is the complement of the clinical hypothesis. So now we split up the heat map into two sets of signals. One involves what the clinicians thought, one involves the electrodes and regions that they didn't think were epileptic. We're going to then feed this into an ensemble of decision trees to predict then how likely was this subject to be seizure-free. And we're going to compare this actual outcome with the actual patient outcomes for 91 subjects. So what does this mean? Well, predicting surgical outcome using these heat maps, conditioned on the clinical hypothesis, will measure importance of neurofragility as a biomarker for this seizure onset zone, or EZ. So we proceed with a 10-fold cross-validation experiment here, where we split up the data or the subjects into different folds or sets of data from training and testing, where we develop the model on the testing set of patients, then we predict and validate on the testing set. And we repeat this process 10 times. And we're interested in one metric, for example, AUC, which is just a statistic that measures model performance. And using 10-fold cross-validation, we can estimate a confidence interval for this statistic. So here we'll show a ROC curve where we have sensitivity on the y-axis, or the ability to predict a successful surgical outcome correctly. And on the x-axis, we have a false positive rate, or how likely are we to predict a success incorrectly. And so an ideal biomarker for the EZ would be on the top left corner, meaning they can predict with 100% accuracy. So in the blue line, we see fragility with the highest AUC of 0.88. And then the other three lines indicate other baseline representations of the data from basically their frequency band power ranges from beta rhythms to alpha rhythms to gamma rhythms. And so we're doing better than those, even within our confidence interval. So to summarize that, we actually showed the AUC now across for fragility in red versus a similar setup for 20 other baseline features that are commonly used in EEG, from frequency band power to graph metrics that I talked about earlier. And even looking at precision, which is just another statistic for model performance, we're also doing better consistently across these different folds. If we also compare neurofragility prediction values, we can see that they correlate in an expected way that we didn't really optimize for, which is good to see. So for example, if we look at the model's prediction probability of success for every single subject, that's on the y-axis here. On the x-axis, we'll see a stratification of different types of epilepsy, ranging from simplest to most difficult, where 1 is lesional, 2 is temporal lobe epilepsy, 3 is extratemporal, and 4 is multifocal. And we see this sort of decreasing trend, which we didn't really even optimize for. So our model is less confident that the patient was going to be seizure-free in these more complex scenarios, which matches what actually happened in real life. If we also now stratify actual outcomes instead of just success and failure. So in the middle plot, we see angle class, which is a clinical measure of outcome, where 1 indicates seizure-free, and for our cases, simplistically, 2 to 4 indicate there's still recurring seizures, but at different rates. And we also see this sort of decreasing trend as well. And we also see the same thing with ILAE, which is also just another measure of outcome. We also see that fragility doesn't really correlate directly with any single frequency band. And so we look at delta, gamma, alpha, beta, high gamma, and in every single window, no single value of fragility directly correlates with another value of a frequency band, power in a frequency band. And that's probably because neurofragility is a network-based measure, whereas these power in certain frequency bands are just describing phenomena in a single EEG electrode. In addition, we can show that neurofragility is actually more interpretable in a certain sense. So in this case, we see a heat map. And if it's a successful surgical outcome, we expect to see a lot of red, right? That overlaps with what the clinical hypothesis. And so we see that in this heat map, normalizing the same way. Now, if we say, take a look at the beta band power map and take a look at the exact same regions, we don't see as many extreme values in the activity. So it's not as visually separable, even though, for all intents and purposes, we try to compute everything the same way and normalize exactly the same way. And so there's not as much red as you would see. However, for our experimental design, decision forests can easily parse out these small numerical differences, even though for us, visually, it's hard to distinguish. And so if we compute an interpretability ratio in this sense that quantifies this, we also see that fragility has the largest effect size. So when we computed fragility, we compared across different subjects with different number of electrodes. We're interested in now kind of trying to interpret this model. So in that sense, we split up the heat maps into two sets of signals in order to facilitate comparison. One was part of the clinical hypothesis, or the SOZ. The other one was the complement of the clinical hypothesis, or the SOZ-C. So we can do a permutation test such that we can actually back out from this model what was most important in predicting the surgical outcome. So in this two heat maps, we see the mean of the importance values over time locked to seizure onset marked in the white dashed line, and the standard deviation on the right. So what does this mean? Well, high fragility near the clinically annotated clinical hypothesis and in the high values in the complement are important. So if you see high values of fragility where the clinicians hypothesize, then it should tell you, okay, this overlaps with what the clinicians thought, and so they're removing fragile regions. If you see, and so then possibly the patient will become seizure-free. If you see high values in the complement, then it wasn't really included in the clinical hypothesis, and so this might be indicative of the fact that the patient was not going to be seizure-free and predictive in that way. Here, we also have an interesting N=1 causal case study, where this was a patient that underwent ablation at the Cleveland Clinic. And initially, the clinicians thought that they should remove regions around X'13. And so in the heat map, we see, okay, it's like a pretty fragile region. But actually, the patient was still seizing afterwards. And so they actually took this patient back into the OR right afterwards and removed the area around L'3, which in our heat map shows that it's even more fragile than before, than X'13 area. And actually, the patient ended up becoming seizure-free in this case. And so that's actually a really cool case study where we had that intervention data in real time almost. So I talked about this retrospective study, and hopefully now you're convinced that, okay, there is some validation. So next we sought to further validate neurofragility in an interesting data set collected from the Hospital of Sick Children. So we tackled the following problem. Once patients obtain surgery, typically, they have to wait at least 1 year to obtain outcome measurements. In addition, it requires observations of seizure data. So what if we could take intrarectal data or non-seizing data, and we could also measure neurofragility after resection and immediately estimate the outcome. That way, it removes this problem of having to follow up with a patient for so long and poor outcomes in general. So we proceed to demonstrate this by first leveraging the virtual brain, or TVB, which is a neuroinformatics simulation platform to simulate realistic EEG data. So we specify a population model of neurons and use subject-specific neuroimaging. And we can then simulate realistic EEG data in this sort of subject-specific virtual epileptic brain, if you will. We can then actually validate these hypotheses on real data where we have access to a subject's pre and post-surgical EEG recordings and right after surgery. And moreover, we can do this with interictal data, that is between seizures. And so we hypothesize that neurofragility will decrease with successful removal of the epileptogenic region. So our original retrospective study assumed a certain structure in the system. And so without going into too many details, I'll just give you a sense of what we did differently in this case. Our original study looked at column perturbations of the system. In this study, we're going to extend neurofragility a little bit further. And we're going to also look at a row perturbation, where our metric of interest is now looking at fragile regions in the sense of both types of column and row perturbation. And so if there exists fragile regions in that sense, then we hypothesize that the EZ is not fully removed. And so now I'll tell you about the virtual brain simulation and how we generate hypotheses. So because it's a virtual simulation, we have explicit knowledge of where we place the EZ in simulation. So those are highlighted in the red electrodes. And then we could simulate a resection where that's highlighted in the orange circle. And we can simulate pre and post resection and compute neurofragility in that sense that I just described and show this heat map. We can do this for a partially failed resection where we don't fully remove the EZ. We can do this for a fully failed resection where we're not even close. We can then quantify how different is the post and the pre-resection heat maps. And we can leverage some statistics to basically do a quantification and comparison of how much fragility decreased after the resection. And so again, neurofragility should have of pre minus post should have a large effect size for successful outcomes, indicating that neurofragility decreased when you remove the EZ. So to describe this bootstrap procedure, we take contiguous chunks of time in small windows as such, and we compute this effect size measurement. And we'll repeat this process, which is known as bootstrapping, to now estimate an effect size statistic with a distribution. And so each dot in this plot represents one sample from that procedure. And then we now have a distribution of this effect size. And we can do this for the successful resection. We can repeat this for the partially failed resection. We can then repeat this for the fully failed resection. We can then plot all these together. And on the x-axis, we see increasing effect size measured by Cohen's d. And on the y-axis, we see different resection scenarios. And so as expected, we see neurofragility decreases after a successful resection of the EZ in the simulated environment, indicated by the high effect size. And so now we can validate these hypotheses, not just on simulated data, but on real data collected at the hospital for sick children. So we can have EEG, so again, this data set is a little bit rarer, but they do a similar clinical procedure where they have electrode implanted, several days to weeks of monitoring. They have pre-resection snapshots of EEG data that they use to formulate their hypotheses. They do this resection, and then they also now get a snapshot of the data during the post-resection period. And then they also typically follow up on the outcome data for at least 1 year. And so this resulted in 6 subjects, pediatric subjects from the hospital of sick children. One had a failure outcome where they still had recurring seizures. The other 5 were seizure-free. So we can take a look again at some representative examples. So here we're showing a failed surgical outcome where the resection is indicated by this orange circle, and then overlaid on the implantation map is the median values of neurofragility during the post-resection period. And so we see some like orange values indicating like relatively higher fragility. Next, if we look to the bottom, we see a pre-resection to the left and post-resection to the right of neurofragility. where some electrodes are obviously removed. And here, everything's normalized the same way, and we see some highly fragile regions after the resection, which is interesting to see because this patient actually ended up still having recurring seizures. If we look at a successful outcome case, we this time see in the heat map that it's a lot more blue, so indicating that there was a decrease in fragility potentially. So we can quantify this in the same exact way as we did with the virtual brain, and we can plot this effect size again in a similar way, where E1 is a subject with a failed outcome, and E3 through E7 were the ones with successful outcome. And Cohen's d, or the effect size, is on the y-axis, and with the dashed line at 0 indicating sort of no change between pre and post resection. And so we see that E1 actually had an increase in fragility after the resection, whereas all the rest had a decrease. And we can also quantify this statistically using p-values, where we did a non-parametric MANOVA, where lower p-values indicates more statistically different. And we can also apply the same sort of experimental design, but this time we look at high frequency oscillations, or HFOs. And so earlier I spoke about this metric or prior approach to localizing the epileptogenic zone. And it's an exciting area because it can be applied interictally. And we don't see the same trend that we would. And so if some marker was really a biomarker for the EZ, for example, then we should see sort of this perfect trend separation, ideally. All right, and so now I talked to you about a retrospective study and an interesting data set with the hospital sick children. And so to end this talk, I'm going to talk about some additional projects that are exciting, that are next steps. So first off, I'll talk about EZ-Track, which is another piece of work that was really exciting that occurred during my PhD. And it involved a lot of engineering, testing, product design that resulted in a PERSIST integration and FDA clearance. So typically, we start off with EEG data, and we then want to hypothesize where the EZ was, right? That's the whole problem of this PhD talk. For us, we want EZ-Track to then be able to incorporate this data and produce a simple to read heat map in just under 30 seconds that can then assist surgical resection. This is done in collaboration with Bobby Norton and Patrick Myers, and involved a lot of open source and open software technologies, which are really exciting to learn about. And this, again, resulted in FDA 510K clearance recently. And we also integrated into a PERSIST platform, which is an EEG software company. Next, we also began looking at the problem of neural correlates for epileptogenesis, which is how epilepsy actually manifests. And in this case, we don't have access to human data, but we have access to animal data, where a colleague in the lab has developed an in vivo rat model with epilepsy. And so we can study the onset of epilepsy in various cases of acute versus chronic epilepsy. This is done in collaboration with Danielle Ahrens and Rachel Smith. And so we have a rat that we have implantations, and we can look at fragility as a marker for the epileptogenesis, where we can look at fragility over time in the baseline period where we know the rat is healthy. Then we cause an acute injury to the brain such that we know they will eventually get epilepsy. And we can track this time period known as epileptogenesis. And then we can also look at epilepsy when the rat is actually seizing. So there's some preliminary data that is exciting because we think maybe this can also further validate neurofragility as a marker. And finally, I did a little bit of this thing called manifold learning with random forests, which is exciting, done in collaboration with Ronan Perry and Chester Quinn and Joshua Vogelstein's lab. And here, to be more simple, manifold learning just implies we want to learn on structured data better, learning on images or time series in such a way that we can perform predictive tasks better. And so in a simulation setting, we can demonstrate that our approach learns significantly better than a suite of other machine learning algorithms. even, for example, a convolutional neural network, because here we're dealing with maybe 1,000 samples. And so we can actually apply this on raw intracranial EEG recordings and demonstrate this, that it can decode movement directionality relatively well in a number of subjects and compare this across different classification algorithms. For example, including convolutional neural networks, which did the worst in this case because you only have around 100 samples. Finally, I'd like to just tell you a little bit about our tool development. So over the years, I've had the pleasure of becoming involved in a lot of open source efforts that I hope will last beyond my time, that are all important in this problem of epilepsy and analyzing neural recordings. I've also become a member of BIDS, which is an open source community-driven standard for storing neural data to share it more easily across laboratories. And I've also been involved in a lot of big Python packages, which have been really a pleasure to learn from, because there are collaborators from all over the world, from France to Asia to Africa and such. And yeah, I want to thank all my funding agencies like NSF, ARCS,